Please join me in welcoming to the Distinctive Voices podium, Dr. Frances Arnold.
Thank you. Thank you. So, I'm thrilled to be here. I build new biological objects by breeding them in the laboratory, and I'm going to tell you about that tonight. And I do it for fun and profit. And I'm going to try to give you a flavor of both aspects of that. And one of the fun things I do is get to come out and talk to audiences like this. And I know you're not supposed to ask questions during the lecture, but this shouldn't be too formal. It's a bright audience. You remind me of my Caltech students. You're here because you're interested in it. It's not a required course. And the whole idea is to see what some of the really fun things that are happening in science and technology is. So I'm going to tell you a little bit about evolving or breeding molecules in the laboratory. So I'm an engineer. I'm inspired by biology. I'm inspired by the marvelous solutions to all sorts of problems that biology has come up with. Solutions to being alive in a challenging world and to being beautiful and functional at the same time. To me, biology is the very greatest of all the engineers, and we can learn a lot from studying biology. We can also solve human problems by engineering biology, by building new biological systems to solve human problems. All right, I'm going to simplify it down. All this functionality that's exhibited in biological systems is encoded in the DNA that's inside the cells. The DNA tells the cells what to do at a particular time, how to respond to their environment, how to develop into an organism, how to convert one form of matter into another, how to extract energy from the environment, and do it with a high efficiency. A number of things we would like to be able to do as engineers. Over my lifetime as an engineer, I've seen almost the entire genetic engineering revolution. The structure of DNA was solved just a couple years before I was born. But I got to experience the excitement of understanding how the structure of DNA contributes to its function and how we can manipulate that DNA, putting new codes into organisms to reprogram their behaviors. This field has exploded. It is such an exciting field to be in. And one reason for that explosion is an explosion in the technological capabilities. For example, sequencing. You've all heard about the fact that we can now sequence an entire human genome for a few $1,000s, and personalized medicine is going to be in your futures in the not too distant future. Alongside with this sequencing capability, that is the ability to read the code of a vast array of organisms, comes an ability also to synthesize DNA. We can build DNA codes. In fact, when you think about it, we can build them from non-living materials. You can take the DNA that encodes an organism, and, for example, from a database, and you can chemically synthesize the entire genome of an organism. It is so fast to do this that I can sit down at my typewriter, don't even have one of those anymore, my new equivalent, a computer, email that sequence off to a supply house, say in Northern California or Korea or South Africa, they're all over the world now, and they will synthesize from chemicals the gene that I have typed in, the sequence that I have typed in. This is one of my students doing exactly that at his computer, and it's now gotten to the point that in a few days you can pretty much order and purchase any DNA sequence you want. Provided it's not smallpox. If you try to type in smallpox, Homeland Security will be knocking at your door. But any other number of things you can buy. So you probably saw this headline last year. This is one of the remarkable examples of how this technology has moved. The Craig Venter group down in San Diego synthesized the entire genome of an organism. A kind of simple, rather uninteresting organism, a bacterium, a simple bacterium. But he synthesized the entire genome nonetheless, 1 million bases of synthetic DNA. And when he put that genome back into a cell whose genome had been removed, degraded, that cell rebooted. It started reading the DNA as if it were its own, and it started dividing again. And it exhibited the properties that were encoded into the new DNA. So that shows, he claimed it was synthetic life. Many would beg to differ. It's not exactly synthesizing life because he had to use an empty cell. But this was truly synthetic DNA made from non-living materials, at least at the DNA level. So this was a great technical feat. Now, what he didn't do that I consider the most important is he didn't write that genome. He merely read it, right, from the organism that had made it, the product of 4 billion years of evolution, and then retyped it in the synthetic material. He did not compose the beautiful music of the genome. And this is really the problem that I'm going to talk to you about. How do you write new biological music? In particular, biological music that will solve a human problem. That's the kind of composing that I would like to do. And we're just learning how to hold a pencil when it comes to doing that. We're very, very good at writing it out in chemicals. If you know what sequence you want, we can play it, so to speak. But we don't know how to compose it. This is a fundamental problem that we have to face. So that's what this new field of synthetic biology, we call it synthetic biology because genetic engineering kind of got a bad name. Synthetic biology, well, we'll see if we can get a bad name for that. But right now it has a good name. We're going to synthesize new DNA that encodes new behaviors in organisms, and it's going to be more than making just a single protein, even though everything I'm going to talk to you tonight is about making a single protein. And I put this particular slide up just to show you that this is fun. This is a field dominated by young people. Kids in garages, kids at laboratory benches, kids on the internet. And they're delighted at their ability to program bacteria to smell like bananas or to act like a photographic plate. But that fun that they're experiencing right now in building all sorts of new biological organisms is going to quickly turn into building things that are useful and important for our world. And in particular, what I'm interested in, and where I think one of the first applications of synthetic biology will come in fact, I know these applications are coming now, is convincing microbes to stop doing what they do best. What do they do best? Make more microbes? They're really good at reproducing themselves. What I would like to do is convince them to become the factories of the 21st century, because what they're great at is extracting energy and chemicals from their environment and converting them into something very complex, a new microbe. Imagine if you could convince them to do the same thing, and instead of making a new microbe, make something less complex. So, for example, a gasoline substitute, or chemicals for making tires, or battery components. But what we could do with this kind of capability is utilize renewable resources. For example, sunlight and carbon from the atmosphere. If you could extract carbon dioxide out of the atmosphere in sunlight, that's what plants do, right? If you could convince microbes to do that and then convert that into, say, gasoline substitute, you'd be in much better shape than we are right now, pulling all that material out of the ground. So I would like to see a whole chemicals industry of this century based on renewable resources via synthetic biology and reprogramming microbes. This ain't easy. And it all comes down to what I alluded to before, and that is that microbes have figured out how to do the problem of reproduction pretty well. And they've been doing that under the force of natural selection for a very long time. So if you're asking them instead to make a gasoline substitute, that's a highly non-natural problem. It's a highly non-natural thing for that to do. And its code, inherent in the microbe, is antithetical to what I want to program it to do. So we have this fundamental problem that, yes, we can synthesize new code. We can insert it into the cell. The cells will start reading it because we know some of the signals that they need to start reading that code as their own. But we don't know what sequence of these little 4 letters of the DNA code will program this kind of behavior. So what do we do? I told you I was going to talk about the simplest problem, really, in this, which is making new proteins. And that's not a simple problem at all. And I realize many of you are not biologists, so I'll give you a few little graphics here to illustrate the problem that I'm talking about. Cells are made up of many components, but some of the most important components are these molecules called proteins. And the ones that I like catalyze reactions that convert one form of stuff into another form of stuff, hopefully stuff that's more valuable than what you start with. And so this chemical catalyst is a very complicated molecule. And it's encoded by the DNA that codes for it. That DNA gets translated into a protein sequence. So we understand the mapping from this DNA sequence to the protein sequence. That's the genetic code. We've known that for many years. So we understand that. But what we don't know is how this DNA code encodes this folded structure. Now, I've shown this little red thing over here because that's our artist's rendition of the more complicated 3-dimensional X-ray structure, for example, showing all the atoms inside of a typical protein. This shows how the polypeptide chain, which if you stretched it out would be a long piece of string, folds up into a 3-dimensional structure. And this structure is unique to the protein. Now, this structure is, we don't understand that mapping. That's the so-called protein folding problem. Some of you might have heard that that's a really important problem in biology. Well, you know what? That was an important problem when I was a graduate student. And they told me it would be solved in 5 years. They lied. That problem has not been solved. And it's a very complicated problem because there's lots of ways that a polypeptide chain can fold up into different structures. And they're all subtly different from one another. We don't have the tools or the understanding to distinguish between those different possible states, nor, of course, to search those states. But you know what they didn't do? They didn't tell us how this structure, dictates the function. That's really the important problem, is not the protein folding problem. That's a problem, a straw horse that they put out there saying to students, solve the protein folding problem and you'll get rich. No, you won't. You might get famous, but that won't last you very long. The protein folding problem is a subset of the problem of how does the DNA code encode function. And nobody even talks about that problem because that's really, really hard compared to the protein folding problem. So what do? This is a problem, however, that's been solved before. It's been solved by the algorithm that made all of us here today, that algorithm of evolution. And that's an algorithm that's a very powerful thing that could be used to forward engineer biology. Let me just tell you a tiny bit of evolutionary biochemistry. If you go to the databases today, all these people who have been sequencing genetic material and depositing those sequences in the databases, you can find families of molecules that are clearly related to one another through mutations. You can build family trees of these, just genealogies, basically, of proteins, so that you know that all these proteins that exist today came from a common ancestor that looked just like it a long time ago. And you can identify the mutations that occurred through that process. From these sequences, we know that these this thing is not working. I'm going to put it down. From this thing, we know that these proteins have similar 3-dimensional structures from a given family, but they've diverged in their sequences. The details of their sequences are different, and the details of their functions are different, and they're all beautifully adapted to their particular biological roles. And this process is one of random mutation and the force of natural selection. Do you contribute to the survival and fitness of the organism that makes you? If you do, I'll take your mutations. If you don't, if you can't compete, you get washed out of the process. And the net result is organisms have diverged over evolutionary time. So have the sequences that are different proteins, and so have the details of their properties. Natural selection has solved the protein design problem and done it very, very well. Now, this is an algorithm for design that humans have, in fact, been using for a very long time. For thousands of years, we've been modifying the natural world, right? From corn to carrier pigeons. The influence of human selection, artificial selection, on the natural world is apparent. And I show pictures of dogs because you can see in just a few generations, maybe 500 generations of breeding dogs, has taken you with not too much genetic changes through a very large phenotypic space. These are all dogs. They look quite a bit different for one another. They have different personalities and they have different details of their construction. So this algorithm, humans have practiced it on purpose, choosing who parents the next generation, choosing which progeny get bred and which don't, choosing who lives and who dies, so to speak, in order to create new biological entities. The problem with this is it's too slow for my purposes. And it's too slow because the natural mechanisms for introducing genetic diversity, right? The basis of evolution is the introduction of underlying genetic diversity, mutations. The natural mechanisms for that, that is the underlying error rate in biology, is pretty slow, is pretty low. You can speed that up by feeding mutagens to your children, for example, or radiating them. You can speed up the rate of mutations. Not generally a very good thing to do if you're trying to create smarter organisms, but every once in a while that does happen. And then, of course, in recombination in sex, you can choose who has what with whom and try to control some of the breeding process that way. But it's still somewhat limited. The great thing now about synthetic biology and our ability now to build any DNA sequence we want is that these natural mechanisms no longer become our limitation. I can now dial in any mutation rate I want. I can have sex with 29, 43, 3 monkeys and worms, fungi and archaea, you name it. I get to decide now how the molecular diversity is created. Of course, that opens up a lot of possibilities for failure, right? The reason that our progeny are generally functional is that when you recombine human DNA, you generally get functional human-like DNA. You know, you got 2 eyes and 2 ears and 2 hands most of the time. But the nice thing is that you get a distribution of details in the properties in those progeny. When you start making more dramatic changes in the DNA, most of those become failures, and it's really an open scientific issue. What becomes your opportunity for creating something new and useful? So, evolution is, in fact, hard. Nature figured out the circumstances under which it works in the natural world. I now have to decide how it's going to work in my laboratory. I have to make decisions. I've got my sticky fingers on the evolutionary wheel now, and I have to decide how those mutations are made, who recombines with who lives and who dies. And it comes down to a combinatorial problem. When you think about all the possible ways that you can string together the amino acids that make up even a single protein. I mean, think about it. There's 20 letters in your alphabet. A protein may be 450 letters long. That's hundreds of universes of single examples of those particles. If you close packed them in one copy, you'd have 85 universes full of proteins. So this is a number that's not even conceivable, right? It's not a physically meaningful number. It's, when you think about it, nature never even explored the tiniest fraction of the possible space of proteins. Now, I've explored a much smaller fraction than that. But I can tell you a few things about that space. First of all, it's big. Second, it's empty. Not empty. We're here, right? It's not empty, but it's mostly empty. Most of the ways that you could put together the amino acid sequence of a protein wouldn't encode anything interesting at all. So the problem becomes one of how do you do a molecular optimization process. That's what this is. This is an optimization in real physical molecules. How do you do that and actually create something new and interesting when most of the things that you could make are garbage? Well, it turns out that the answer to that, at least one answer to that, is fairly simple. We have been gifted with rare functional sequences by evolution. You can literally scrape from the bottom of your shoe some of those rare functional sequences. We're surrounded by them. 4 billion years of natural selection has created a whole lot of functional sequences. Even though, on average, the space is empty, we have lots to play with. And not only that, from the experiments that we've done and that nature has done for much longer than I've done, we know that if you make mutations in natural sequences, often they too will be functional. They may differ in details, but they will fold and function in predictable, at least in ways that could give rise to something useful. So that means our fitness landscape, that's what we call this, fitness landscape over the sequence possibilities is rather smooth. And this means that we can use simple optimization processes to explore this through evolution. One mutation at a time. That shouldn't surprise you because that's how natural evolution works. And that's how evolution in the laboratory works. So how do you actually do this? You take that rare functional sequence that's not so rare because it sits on the bottom of your shoe. I work with soil microbes, solve lots of interesting chemical problems in order to survive in the soil and eat the things that it finds there. You can take DNA that encodes proteins from those organisms, copy them with kits that you can buy in molecular biology labs for polymerase chain reaction, and you can dial in a mutation rate. So working with this DNA, I can control how many mutations get made, and I can make a lot of different versions of these genes. Then the cells do the hard part. This is standard genetic engineering. You put this DNA inside a microbial cell. It'll start reading it as if it were its own, but it'll now start putting in mistakes in the proteins that are encoded by the mistakes in the DNA. So you can make lots of mutant proteins that way. Now comes the hard part for me, because I have to sift through all of these different proteins and decide who is starting to acquire the properties that I'm trying to breed into this system. So if I want something that's more stable or more active or convert something into something else that I like better, I have to measure that property using analytical chemistry tools that have been developed for hundreds of years. We can do that, and we can, for example, convince the protein that if it does a good job, it's going to give a deeper purple color in my assay plate. And if I've asked the right kind of question, an evolutionary question, one that can be solved by 1 or 2 amino acid substitutions, I will find that some of my mutant proteins are better, and then I can iterate on this, walking uphill in this evolutionary process. Now, of course, the vast majority of mutations, as I told you, are deleterious. Most things are not better than what you started with. Those just get thrown away in this simple process. So it becomes a random uphill walk in this fitness landscape. But many times, this simple process works very, very well. We're adapting the protein to its new jobs. What does that allow us to do? We can make things that nature never cared about. Biologists go, who cares, right? But I want to make things that will solve human problems. And I guarantee you, you need to be outside of biological space a lot of times in order to do that. I also love to tweak biologists because I relegate them to this little tiny spot in the sequence space. Because only a very limited number of molecules are relevant to biology, survival, that contribute to survival and fitness of an organism, right? All the other ones, for example, the cure to cancer or the solution to the energy crisis, are out there solving problems that nature doesn't care about. They're out here in my physically possible space that I, as the engineer, want to explore. And this is my sandbox. This is where I get to play. And being able to walk between biology and the physically possible tells me deep things about what biology actually cares about versus what physics dictates. It's a fun place to play. This process works very well. This is a very colorful example from my friend Roger Chen, who collected green proteins, green fluorescent protein from jellyfish, and red versions from a red jellyfish, and proceeded to evolve them in the laboratory to display a whole palette of beautiful fluorescent colors. Of course, they go into making fluorescent goldfish, and you won't win a Nobel Prize for doing that. But he won the Nobel Prize because not only are they good for making colored cats and colored goldfish, they completely revolutionized the way we do biology. We can now label proteins inside of cells and see exactly where they go and where they're expressed. And this has just had an enormous impact on our understanding of biology at the molecular level, rightly winning him the 2008 Nobel Prize. I wish I had done that, but I didn't. I'm working on much less colorful things. I'm trying to make catalysts that convert one form of stuff into other forms of stuff. And I know not too many chemists in this group, and I promised I wouldn't show lots of chemistry, but you're just going to have to sit through this. This enzyme is so cool because it inserts oxygen into an unactivated CH bond at room temperature and pressure. And if you were a chemist, you go, oh my gosh, that's just amazing that a molecule can do that. That is really incredible. And actually it is, because then with a molecule that can do that, you could do things like take natural gas and convert it to liquids. Because natural gas with an oxygen in one of those CH bonds becomes an alcohol. You could drink it too, but you could drive it. You could take these gaseous resources that we usually flare off on oil wells and convert it to a liquid fuel form. That's like one of the holy grail problems in chemistry. And so I'll just point out that this is a really complicated molecule that does that. We like to use these big globular structures, but they illustrate complicated protein molecules that do that. So I work with this, and the only problem is the one that I can scrape off the bottom of my shoe, yes, it does insert oxygen into a CH bond, but it doesn't do it with alkanes. It does it with fatty acids. Nobody cares about putting it there. So, one of the questions we could ask, could we evolve this enzyme into something that instead of doing it on a fatty acid, we could breed it to take it down to something like methane? That would be lovely if we could do that. So this is actually a more interesting problem than just the practical problem, because nature doesn't use this particular version of this enzyme to do this chemistry. Nature has a whole other set of enzymes that does this chemistry. There are organisms that live on methane and ethane, but they're a whole different mechanism for doing that. And so there's no known member of this family that does this. And some people told me, oh, Francis, it can't be done because this mechanism is not chemically competent to insert that oxygen into that bond. That's possible, right? But it's also possible that we don't know of any P450 that does it because microbiologists don't like to work with alkanes and oxygen. They tend to blow up. And microbiologists are not really good with things that blow up. So there may be other reasons that we don't know a P450 that can do that. So this is exactly the kind of problem we can test by evolving things in the laboratory. We can ask, is this molecule competent at doing that? If it is competent, then I can guarantee you somebody is going to find one out in nature. All right. So the answer is yes, of course, it is chemically competent to do this. You can breed this molecule over multiple generations, first addicting it for activity on octane on the C8, and then moving it up with time over multiple generations until you make an enzyme that is essentially as good on propane as it was on its native substrate. It's perfectly happy to insert oxygen into propane. And we were very pleased to show that this molecule exists. And not only does it exist, it's only got a few mutations, 23 out of 500, well, actually out of more than 1,000 of its natural amino acids. In fact, these kinds of studies have shown multiple times that enzymes are highly evolvable. They adapt, and they adapt rapidly with just small changes in their sequences, maybe 1 or 2% of the sequence, and you get a completely new version of the molecule that's now really good at doing some other kind of job. Here's the scary part of it, though, and this is what really bugs the would-be rational designers of molecules. There's a whole other camp that says, oh, you know, only engineers would do random mutation and evolution. If you're really smart, you'd sit down and design it from first principles. Well, the scary part is that when you see the mutations that give rise to these new properties, they're all over the protein. And we can't even explain them, much less predict them. So I'm not going to be too worried about the rational designers, at least in my scientific lifetime. Maybe in my students, they'll see some competition from that. Why is this? The reason is that in biology, I'm really sorry, physicists in the group, details do matter. The difference between being alive and dead, you know, it's like details, and that matters a lot. Proteins being unfolded and non-functional versus folded and functional, details. We don't understand those details, and it's going to be a long time before we do. All right, let's look at another search strategy for evolution. I'm fascinated by the enzymes that break down biomass. We'd love to get sugar out of cheap biomass like cellulose, right? You've heard all about this. We're going to solve the world's energy problems by, you know, switchgrass or, you know, municipal waste or growing trees in the forest. The problem is these guys don't like to give up their sugars so that we could feed it to fuel-producing organisms. So we use little proteins that do that, enzymes that'll chew up these polymer chains of sugars and release the sugars for fermentation. And to make new versions of this, we decided to do a little bit of recombination. In this case, taking cellulolytic enzymes from organisms that had evolved to use cellulose, like termites or fungi, breeding those together to create new versions of cellulose that nature has not seen before. And we use computational methods to speed this up. Yes, I'm willing to contaminate my experiments with a little bit of rational design, but it has to be the right kind. And we find that sex improves their attitude a lot. You can make much happier cellulases by recombining them from different organisms. So for example, we make these so-called chimeric versions of these proteins that are much more stable than any of their parents. And when you add random mutations in there and optimize those, you can make versions of these enzymes that are happily working at temperatures that the organisms that make them won't tolerate. And we find that recombination works, in fact, very well, which shouldn't surprise you. This is a much more diverse audience than I usually get at Caltech. I can usually rib my Caltech audiences that are 90% male by asking them, what on earth are you contributing to the survival and fitness of the population, right? Because you're not carrying any children. If the population really wanted to grow fast, right, if that's the criterion for survival, you get rid of all the males. So you have to argue that recombination, sex, gives you benefits that you don't get if everything's parthenogenic, right? Well, I guarantee you there are benefits to sex. You can see them right in the laboratory at the level of individual proteins. And it's just like, you know, when you have kids, sometimes their capabilities lie within the capabilities of parents. Sometimes there's these wonderful extremes. Some are dumber, and some are smarter. And I'm finding those smarter ones. I find proteins that are better than any of the parents they work with. You can see it. All right. So these become more stable. You can heat them up, and they work at much higher temperatures. And when you make things that are more stable and work at higher temperatures, you can actually get benefits in chemistry where the rate of reaction goes up. And so these enzymes, for example, will be much more active at their optimal temperatures than the native enzymes. These are the industrial workhorses now, are at their temperatures. And in fact, we've sold these enzymes to the major enzyme suppliers for use in biofuels production. The net result now, though, with all this kind of technology and the evolution and being able to write the genome now with these new tools, is that we can now retrofit these organisms to become the factories for the 21st century. So right now we make ethanol from sugars, right? Why do we make ethanol? Anybody know why we make ethanol? Wow, we want ethanol, right? You can drink it. We've been making ethanol for a very long time. So humans have been creating ethanol yeast and breeding ethanol yeast for thousands of years. And we have lots of very efficient ethanol producing yeasts out there. So when the oil crisis hits, a few years ago, boom, all that nice ethanol production can go into fueling your gasoline. You know what? Ethanol is a lousy fuel, though. It really is not the right kind of fuel. So if you're so smart, we should be able to retrofit organisms to make something else. And that's exactly what we do. So now with these tools, we can retrofit yeast, say ethanol-producing yeast, to make something like isobutanol. Who knows what isobutanol is? Oh, we got a few hands. That's really great. Isobutanol, that's the perfect stuff. You got 4 carbons now instead of 2. It's miscible with gasoline. It won't corrode your distribution system. It can be made into about most of the chemicals industry. You can dehydrate isobutanol and essentially make any kind of hydrocarbon mixture out of it. So it's your perfect material. And in fact is the basis for whole drop-in chemicals and fuels capability. And that's the basis of the company that I formed in 2005. So this is not science fiction. This is actually reality now. There's 2 plants in Minnesota that are producing isobutanol as we speak. I'm going to use my last few minutes, though, to go off into the future, maybe the distant future. But this is meant to leave you with a sense of where the creativity in this all comes in. We can solve human problems. We know what those problems are. It takes creativity to solve them. There's a whole other group of people that are just exploring what could we do? What could we possibly do, and how can we do it? And one of the really fun things is to think about, well, yeah, Francis, that big space of proteins that you're playing in is a big place. But we could make it a lot bigger by bringing in other materials that nature often doesn't use. So let's talk about other atoms in the periodic table. Now, we know nature does use things like calcium and other minerals and makes beautiful structures from those. And those structures, which you see in diatoms and shells, are encoded by proteins. Proteins template the formation of these minerals and these materials. And since proteins dictate how these things form, you can make new ones by evolving them in the laboratory. So people like Angie Belcher, up there at my rival school in Boston, she plays with proteins that will encode new materials development. And I love this because she uses viruses. She puts viruses to work to do this. She can put a gene that encodes a peptide on the tail of a virus. And when she makes mutations in that, this virus could then stick to these new minerals. And when you've got something like that, then if you just pan these viruses, if you just put the material in a collection of these mutant viruses and you screen for their ability to bind to these materials, and they can be anything. They can be metals and minerals and semiconductors and polymers, whatever you dictate. The ones that stick stay on the material, and then you can wash off all the ones that didn't work and just wash them off down the drain. And you can go through an evolutionary process to discover the proteins that will template these new materials. And that's way cool because then you could put those proteins, like she does, on the coats of the viruses where they'll template those materials and they'll form little nanoparticles of the materials. And those nanoparticles can form basically these nanostructured wires. And just to give you an example of what you might do with that, she made a high-powered battery on the back of a virus that templated an iron phosphate material and also bound single-wall carbon nanotubes. So it bound the iron phosphate on this part of its structure, and then she stuck peptides that bound single-wall carbon nanotubes. And she was able to make a cathode material that gives higher power than any of the more conventional materials that she'd made, all on the back of a virus. And what this does, as I say, is it allows you to expand the capabilities of biology to cover a lot more of the periodic table. Her view is, might as well go everywhere in that and see what biology could do if you gave biology a reason to do that. Well, why stop there? There's other people who are thinking about, well, gosh, 20 amino acids, that might keep you happy. But I'd like to have a lot more letters in my alphabet. The chemists are going crazy here. Because if you did that, why stop there? You could actually think about using DNA not to encode proteins, but to encode any material, any set of monomers, amino acids, you name it. So maybe we could use this whole protein production machinery in a whole new way. And I love this little movie. This little movie actually illustrates what we know about the machinery that reads DNA. So this is your DNA here. And it's reading the DNA and bringing the amino acids one by one according to the code, the triplet codon code that it's reading. And it brings these amino acids on the tRNAs. And it assembles them one by one in this structure. And it's assembling a protein that's going to start coming out this end. So this is a generic machinery that reads a code and translates it into stuff. That's why this is so incredibly powerful. If you could manipulate this to read the code, and instead of bringing in natural amino acids, bring something else, you could have encoded materials coming out the other side of this. Well, this is not science fiction. This is, in fact, what the research is happening now at a number of places, including Caltech and also at Scripps in Pete Schultz's lab. He's manipulated the ribosomal machinery to selectively incorporate non-natural amino acids. And this is just a trivial example to illustrate what you could do. If you put in a non-natural amino acid in this protein, you can break the immunological tolerance. That is, if you put a mouse protein in mice, it won't build up antibody response to that. But if you manipulate the protein structure and incorporate non-natural amino acids at particular positions, so here's a new amino acid that was invented by a chemist and put at these new positions and now immunize the mice with its own protein, it will make antibodies that recognize this protein, but also the native version of the protein. So you can actually make antibodies to self-proteins using this chemistry. All right. As I said, chemists have now gone wild. They have synthesized hundreds of new amino acids. And not only synthesized hundreds of new amino acids, but have evolved, they've used directed evolution, to modify the protein machinery of that ribosome to recognize those amino acids, bring them into the machinery, and use them in place of the native amino acids. So this, I mean, if there are any chemists here, you should be drooling with envy, because now you can see all sorts of functional groups, triple bonds, double bonds that you couldn't incorporate into proteins before, all done according to a DNA fold. But why stop there? Maybe you could do that with non-protein things. So here, I love this experiment. This young fellow at MRC in Cambridge who said, well, gosh, if I've got hundreds of amino acids, now I become limited by the genetic code. It's a triplet code. Four letters, triplet code, you got 64 possibilities. That's way too small when the chemists are going nuts. So why not have a 4-letter code? Maybe I could evolve this machinery to, instead of reading 3 letters at a time, it's now going to read, its codon will be 4 letters. Now I'm just going to put that alongside of the normal ribosomal machinery inside of a cell and have these two sets side by side, one that makes the cell and the other one that makes a protein according to my new genetic code. So in fact, that's exactly what he's done. He's made a so-called orthogonal ribosome that will now read an RNA strand that's been engineered now with a 4-letter code, and he can encode 256 different amino acids on that. So, if you get tired of 256, then you're going to have to move to a 5-letter code, but apparently we can do that. And once again, he used directed evolution to create that, because it's a complicated piece of machinery. So he just put mutations down at the codon recognition site and forced the thing to solve a problem where if it could do it, it would live, and if it couldn't, it would die. And by golly, that molecule learned how to read the 4-letter code. So now we have this whole new protein translational machinery that can take DNA and create a whole new set of polymers that weren't accessible to us before. What does this give us? I mean, this is just an example of the kind of creativity that's going on in this field of synthetic biology. We're expanding the space of materials that we can make biologically, no longer limited to just carbon, oxygen, nitrogen, phosphorus, all those materials scientists there would say, oh, biology is weak, it's very slim pickings over there. Now we can actually access some really exciting materials and some really exciting chemistry that we couldn't do before. And it all becomes evolvable then, so your space of possibilities grows rapidly. Well, that little bit of view into the future, I want to thank you for coming tonight and listening to this, and I especially want to thank Mr. Darwin, without whom this would not have been possible. And we are here to enjoy the products of evolution. So thank you.